CodeOcean apps include a selection of ready-to-use bioinformatics tools. CodeOcean also includes functionality that lets us transform our computational work into no-code apps for others to use. These include Shiny, Streamlit, and our built-in parameterization tool, the CodeOcean App Panel. In this demo, we will walk through the pre-built app library, CodeOcean app panels, and an example of a capsule running Shiny. So here we have a selection of bioinformatics tools that we are regularly adding to. These tools include data connector capsules, various genomics and transcriptomics applications, visualization tools, and more. So the first example we'll cover, let's check out the Max peak calling capsule. This is a tool to identify transcription factor binding sites in ChIP-seq data. We can take a look at the README file that is provided by CodeOcean. So here we can take a look at the inputs, including the comparison sheet as well as the alignment files so that we know which data to analyze. We can also take a look at the app panel parameters. So there is a detailed description for every single parameter that is to be used in this capsule. We also have a description for all of the output files that you can expect from this analysis. And of course, we provide the original source of this Max tool. On the left-hand side, you can see an example of the panel itself. Now, this is a read-only version of the panel, but we can see on the left-hand side here the data that the user will be able to swap out for their own data once they have duplicated this capsule for their own use. We can also see all of the parameters. Some of them are drop-downs and some of them are open text fields. These are all of the parameters, and most of them are optional for this tool. And lastly, on the right-hand side, we can take a look at the timeline for this CodeOcean apps capsule. Right now, we are looking at version 2.0 of this capsule. If we wanted to take a look at an older version, we can also switch to that version as well. Every version in the timeline, you will see the release result that is associated with that version. If we hop back to the latest version 2.0, we have the option to duplicate each version. So I'm going to go ahead and duplicate the latest version of this CodeOcean apps capsule. Go ahead and confirm that. And now we are in the process of duplicating this capsule from the CodeOcean apps library into my dashboard for my own view. Now, as the duplication continues, I want to show another example that I have ready to go here. This is another example of a CodeOcean app panel. In this case, this is associated with a pipeline. So let's get the full view of the pipeline here. This is a visual representation of this RNA-seq quantification pipeline. We can see all of the next flow processes that are represented as CodeOcean capsule. On the left-hand side, this should look pretty familiar. This is the app panel itself. In this case, we have 3 different data that we can either use as is or swap out for another data asset. So we have the sequencing reads here. We've got the star index, and we have an annotation file. Now, any of these we can substitute with our own data. So just as a quick example, I'll swap this out for sequencing reads. And if we now scroll down to the parameters, clicking on any parameter, we will see highlighted the exact capsule that is associated. In this case, we are looking at the star alignment. Any changes that we make, we'll see a little dot representing changes from the default. Let's say we want to reset. There's an easy option to do that, to reset to the default parameters. You can see here all of the parameters that are associated with every step, every process in this pipeline. And on the right-hand side, just like before, we have a timeline for this pipeline. We can see the results that my colleague Steven ran here. We can also swap around to different versions of a pipeline. Right now, we're looking at release version 3.0. Again, every result that is associated with any given version can be found in the timeline of the capsule or pipeline. Now let's take a look back and see if our CodeOcean apps capsule has finished duplicating, and it looks like it has. Let's go ahead and see our new personal private copy. And here we go. We now have our own copy of Max peak calling. We can swap out our data. We can modify any of these parameters. Click run, and we will be off and running. The last example that I want to show is going to be a no-code example using our Shiny. So I'm going to hop over to no-code apps here. And here I've got a Shiny cell example. let's go ahead and hop into Shiny. This Shiny cell capsule is dedicated to 10x single cell RNA-seq analysis. We can take a look at this application view that hides the code from the user. We can use this tool to analyze and make figures for the single cell RNA-seq analysis. And when we're ready to capture results, we'll find this image stored in the timeline of the capsule. Just to hop around and show the application view, a lot of different tools here that we can use to analyze our data. I'm going to go ahead and shut this down now. The last thing I want to show is hop into the Shiny capsule itself. So here on the left-hand side, we can see the actual files that make up this no-code application. So here's the server file, as well as the UI file. We can see that this Shiny cell capsule is a release version of the capsule. That means this is a capsule that is currently in production. If we take a look at the top right corner, the only cloud workstation we can launch is Shiny. No other cloud workstations are available to us. And that's how we know that this is a capsule that is running in production. If you have any questions about how our app's features work, please make sure to take a look at our documentation or book a demo with us to get into more detail.